Now, let us see that how glycogen is formed in our cells. It is a very interesting but little bit complicated process. So, I need your real focus in this part. First of all, you already know, I have told you that glycogen is synthesis from the monomers of glucose. Those are the alpha D glucose monomers. This process occurs in the cytosol and it requires energy, of course, in the form of ATP, and there is a new molecule, that is UTP, uridine triphosphate UTP, which is also used in this process. Then actually what happens that glucose, that is alpha D glucose, it combines with the UTP to form a glucose UDP complex, and the enzyme which is involved in it is UDP glucose pyrophosphorylase, UDP glucose pyrophosphorylase. Also keep in mind that here pyrophosphate hydrolyzes to two inorganic phosphates by pyrophosphatase. This is an exogonic reaction which releases energy. Then until now you have been hearing about glucose 6-phosphate, but here we do not use glucose 6-phosphate to form this complex, but rather instead we are using glucose 1-phosphate. So, from where the glucose 1-phosphate molecule comes? The glucose 1-phosphate molecule actually you obtain from the activity of phosphoglucomutase, an enzyme which convert glucose 6-phosphate into glucose 1-phosphate molecule. If you see here you can see the glucose 6-phosphate is being converted into glucose 1-phosphate through the intermediate which is glucose 1,6-biphosphate, and the enzymes which is involved is phosphoglucomutase. So, in the presence of this enzyme you finally get the glucose 1-phosphate moiety, and this glucose 1-phosphate moiety will be then involved in the formation of glucose uridine diphosphate complex. Keep in mind that the linear chain of glycogen contains alpha 1,4 linkage, and glycogen synthase enzyme is involved in it. But also you should always remember that glycogen, on its own cannot start its formation from a single residue of glucose. You have to have certain residues of glucose first in the form of a short fragment which will be consisting of certain residues of the glucose, and that short fragment which act as a primer. So, once you have that primer of short fragment of glucose 1-phosphate residues which are linked with each other through glucose 1,4 linkage, then the glycogen formation or elongation in the glycogen will really start. But what if you do not have any fragment of those glucose residues? Then at that time what happens that another fragment known as glycogenin, a small fragment of glycogenin is involved in the formation of that fragment. So, the glycogenin molecule is involved in the formation of that short fragment which will act as a primer for the elongation of the molecule of glycogen. Keep in mind that glycogen जो भी फ्रेगमेंट बनाएगा वो 3 से 4 या 5 जितने भी रेजिड्यूज का फ्रेगमेंट होगा, ग्लूकोज रेजिड्यूज का फ्रेगमेंट होगा, वो अल्फा 1,4 लिंकेज होगी. जब हम अल्फा 1,4 लिंकेज की बात करते हैं, व्हाट डज इट मीन? अल्फा 1,4 लिंकेज से मुराद है कि वो एक लीनियर चेन होगी. Once you have that short fragment being formed by the glycogen, then you can elongate that chain to form a full-fledged molecule of glycogen, and that enzyme I have already told you is the glycogen synthase that will be involved in this formation. But again, first you will have to have that fragment in your hand, and then the process of glycogen elongation takes place.